What is the chemical composition of a protein? What is the structure of a protein? What is the nature of genetic code? What is the structure and function of transfer RNA? What is the structure and function of ribosomal RNA? How is polypeptide synthesis initiated on the ribosome? How is polypeptide elongated on the ribosome? How is polypeptide terminated in the translation messenger RNA? How our proteins are sorted in the cell? Changing single letter in a word can completely change the meaning of the word. This term can change the meaning of the sentence containing that word. In living organisms, a sequence of 3 nucleotide letters produce an amino acid word. The amino acids are strung together to form polypeptide sentences. Lecture. We will study the process by which nucleotide letters are translated into polypeptide sentences. One of the most important applications of human genome research is the use of sequence information to track down the cause of genetic disease. Cystic fibrosis, one of the examples. How the genetic code this field? Cell-free protein synthesized machinery isolated from E. coli, ribosome, transfer RNA, protein factor, radio-labeled amino acids. Synthetic messenger RNA contain only one type of base. U, this is the code for phenyl. C is the code for proline. A for lysine. G equal for what? It's unsuitable. Synthetic copy lemma. CCA, ACC, CAA, composed of 2 different bases. Proportion of AC vary to determine exactly which codon specified with amino acid. Synthetic polynucleotide of unknown composition. The Nobel Prize in Physiology or Medicine in 1968 was awarded to 3 scientists, Hawley, Gubin, Khorana, and Marshall Nirenberg. They were recognized for their interpretation of the genetic code and its function in protein synthesis. The Nobel Prize in Physiology or Medicine during the fall of 1868, a young Swiss physician by the name of Friedrich Muscher isolated a new type of compound from cell nuclei. He called this compound nuclein. Today, we call it nucleic acid. 2 years earlier, Mendel in the Tower of Brno finished a series of experiments eventually turned out to be connected with major discovery. From a very simple experiment with B, Mendel discovered that our inheritance is packaged into many independent genes. Mendel marked the beginning of genetics as a science. Nucleic acid and genes, originally 2 widely separated concepts, together from the basis. Initial results were published in Extenso only after his death in 1890. Mendel reported his observation in the first publication in 1866, which received little attention and was soon forgotten. For a long time, no connection between genes and nucleic acid could be seen. In so doing, he demonstrated that genes are made up of nucleic acid. Harvey discovered the marked beginning of the new branch of science which has come to be called molecular biology, and which up to present time has been concerned mainly with the biochemistry of genetics. The genetic code and why it's called the code of the live nucleic acid are very complicated molecules, but their structure shows certain regularities. They are constructed from a limited amount of smaller building blocks. If we compare nucleic acid with a language, we can think of the building block as the letter of the alphabet of the language. With this analog, we may say that the language of nucleic acid in the cell describes our inherited traits. It tells us if our eyes and those of our children. It tells us if our eyes and those of our children are blue or brown, if we are healthy or sick. A brown-eyed child receives from its parents nucleic acid, which have the ability to direct the formation of protein required for the synthesis of dark pigment of the eye. It is the chemical structure of nucleic acid which determines the chemical structure of the protein. The alphabet of nucleic acid dictates the alphabet of protein. The genetic code is the dictionary which gives us the translation of one alphabet into the other. When the hieroglyphs were deciphered, the archaeologists made us use of Rosetta Stone, which contained the same alphabet written with Greek and Egyptian letters. In theory, one could use the same method for deciphering the genetic code by comparing the chemical structure of particular nucleic acid with that of the corresponding protein. Letter by letter, however, this is not possible for technical reasons. A very simple and ingenious solution. He realized that the chemist had a decisive advantage over the archaeologists, since he could construct in the test tube a system which used nucleic acid as a template for the formation of protein. Such a system can be compared with the translation machine, which fed by the scientist with the sentence written in the alphabet of nucleic acid. The machine then translates the sentence into the protein alphabet. What is the mechanism for the translation of the code within the cell? This question was successfully attacked by Holly. Is one of the discoveries of special type of nucleic acid which has been called transfer RNA. This nucleic acid has the capacity to read of the genetic code and to transfer it to the protein alphabet. After many years, Holly succeeded in preparing transfer RNA in pure form, and finally in 1965 established its exact chemical structure. Holly work presents the first determination of the complete chemical structure of biological active nucleic acid. There also exists a second language in our cells, the language of proteins. Written in the alphabet of protein, a single cell contains many thousands of proteins which perform all the chemical reaction required for normal life of the organism. Synthesis of each protein is directed by particular nucleic acid. Research came of age 1944. In this year, scientists have succeeded in transferring a hereditable property from one bacterium to another with the aid of pure nucleic acid, and in so doing demonstrated that genes are made up of nucleic acids. How was genetic code transferred? In process of translation, ribosome binding assay of Nuremberg and Leder in 1964, protein synthesis doesn't occur. Only one type of charge transfer will bind to the trinucleotide single RNA UU with phenylalanine code anticodon, messenger RNA UCU, transfer RNA with serine codon anticodon, messenger RNA CUC with leucine codon anticodon. Identified 50 codon using this method. Combination of different methods eventually identified 61 codon. The other three don't specify amino acids. The first letter, second letter, and third letter. This is a chain termination codon, stop codon, initiation codon, stop codon, UAA, UAG, UGA, the initiation codon, AUG. It's code for methionine. This is code UAA, UAG, UGA. GA. Characteristic of genetic code written as in single RNA 5 to 3. Code is triplet. Each 3 nucleotide coded in a single RNA specifies one amino acid. Code is comma-free. Single RNA is read continuously 3 bases at a time without skipping bases, not always true. Translational frame shifting is known to occur. Code is non-overlapping. Each nucleotide is part of only one codon and is read only once. Codon is most universal. Most codons have the same meaning in different organisms. Code is degenerated. 80 or 20 amino acids are coded by more than one codon. Methionine and tryptophan are the only exception. Many amino acids are fourfold degenerated at the third position. Code has start and stop signal. ATG codes for methionine is the usual start signal. TAA, TAG, TGA are stop codon and specify the end of translation of a polypeptide. What will occur in transfer RNA anticodon. Third base is less constrained and barely specifically. Whipple hypothesis proposed by Francis Crick in 1966 occur at 3 end of codon, 5 end of anticodon, result of arrangement of each bond of base pair at the third post. Degeneracy of codon such as Whipple always result in translation of same amino acid. Complete set of codon can be read by fewer than 61 transfer RNA. Anticodon GCAU pairs with U or C, C pairs with G, A pairs with U, U pairs with A or G, I inositol pairs with AUC. This is identical transfer using transfer RNA. This is the codon and amino acid coded for. Evolution of the genetic code. Each codon possesses an inherited set of possible one-step amino acid changes, precluding all other. As a result, some codons are inherently conservative by nature, whereas others are more radical. Only 12 results of moderate radical or very radical amino acid change. Most changes are nearly neutral because they result in substitution of similar amino acid. DNA sequence with different codon composition have different properties and may evolve on different evolutionary trajectories with different rate of substitution. Evolution of genetic codon. On average, similar codons specify similar amino acids such as single base changes result in small chemical changes to polypeptide. For example, single base changes in the existing code have smaller average effect on polarity of amino acids. The code has evolved to minimize the severe deleterious effect of substituting hydrophilic for hydrophobic amino acid. Translation protein synthesis. Protein synthesis occurs on ribosomes. Single RNA is translated from 5 to 3. Protein is sensitized in terminus to C terminus. Amino acid bonds to transfer RNA are transported to the ribosome, facilitated by specified binding of amino acid to the transfer RNA. Complementary base pairing between the single RNA codon and transfer RNA anticodon. The single RNA recognizes the transfer RNA codon. Translational for main step charging of transfer RNA, initiation, elongation, binding of amino acid transfer to the ribosome, formation of peptide bonds, translocation of ribosome to next codon. Charging of transfer RNA, amino acylation. Amino acids are attached to transfer RNA by amino acyl transfer RNA synthetase, produces a charge of transfer RNA, amino acyl transfer RNA, use energy derived from the ATP hydrolysis, 20 different amino acyl transfer RNA synthetase, one for each AA transfer RNA, process enzyme specific recognition sites, sequence of event, ATP and amino acid bind to amino acyl transfer RNA synthetase to form amino acyl A and B, plus 2 phosphates transfer RNA bind to amino acyl A and B. Amino acid transfer to transfer RNA displacing A and B. Amino acid always is attached to adenine on 3 end of transfer RNA by its carboxyl group, forming amino acyl transfer RNA is a series enzyme catalysis, coupling of amino acid to A and B to form amino acyl A and B. Uncharged transfer RNA bind to the enzyme. Enzyme transfer amino acid from amino acyl A and B to transfer RNA to form amino acyl transfer RNA are released from the enzyme. Amino acyl transfer RNA synthetase and the amino acid and the ATP bind to enzyme. And this is a cycle or the steps. I start with amino acyl transfer RNA synthetase. Step 2, initiation requirement, messenger RNA, ribosome, initiator, transfer RNA, 3, initiator factor, magnesium, guanosine triphosphate, initiation step prokaryote, 30 S ribosome subunit, plus bind to AUG start codon and shine the Lagrange sequence composed of from 8 to 12 purine rich nucleotide upstream. Shine the Lagrange sequence is complementary to 316 S ribosomal RNA, initiator transfer RNA, bind to AUG with 30 S subunit. All new prokaryote protein begin with F-met, later to move. F-met for methionine modified by transformylase. AUG at all other codon positions simply code for methionine. IF3 is removed and recycled. IF1, IF2 are released and the GTP is hydrolyzed, catalyzing the binding of 5S ribosomal RNA subunit. The initiation factor, 30S ribosomal subunit, 30S initiation complex, attachment of 50S subunit, 17S initiation complex. Initiation differences between prokaryote and eukaryote. Initiator is not modified in eukaryote, but eukaryote possesses initiator transfer RNA, no shine the Lagrange sequence but rather initiation factor. Eukaryote AUG codon is embedded in the short initiation sequence called Kazakh sequence. Eukaryote poly A tail stimulate translation by interacting with 5-cap, forming a single RNA cycle, elongation of peptide, binding of the amino acid transfer RNA to the ribosome, formation of peptide bond, translocation of the ribosome to next code. Binding of the amino acyl transfer RNA to the ribosome. Ribosome have two sites, B site and A site, relative to messenger RNA. Senses begin with F-met prokaryote in the B site and AA transfer RNA hydrogen bond to AUG initiation codon. Next codon to be translated is in the A site. Incoming amino acyl transfer RNA bound to elongation factor, hydrolysis of GTP, release another elongation factor, remove GDP and bind another to the next AATT RNA. Cycle repeat after peptide bond and translation. This is the step. And this is complex elongation factor. First step, 70S initiation complex is formed, and the complex with elongation factor and GTP. The next amino acid transfer RNA molecule. Peptide bond forms between the two adjacent amino acids. Translocation occurs. And when translocation is complete and the peptide transfer RNA is in the P site, uncharged transfer RNA is released from A site, and the ribosome is ready for another elongation cycle. Formation of the peptide bond. Two amino acid transfer RNA positioned in the ribosome, one in the B site and another in the A sites. Bond is cleaved between amino acid and transfer RNA in B sites. Peptidyl transferase forms a peptide bond between the three amino acids in the B site and amino acetyl transfer RNA in the C site. Transfer RNA in the A site has the ground peptide attached to the peptide transfer RNA. Adjacent amino acid transfer RNA and the following peptide bond formation. Translocation of the ribosome to the next codon. Final step of the elongation cycle. Ribosome advances on the codon on the messenger RNA. Use EFG binding of charged transfer RNA inside 3 is blocked, and charged transfer RNA in B sites is released. Peptidyl transfer RNA moved from A site to the B sites. Back and A site now contain a new codon. Charged transfer RNA to codon bind to the A site, and the process is repeated until a stop codon is encountered. Number and type of EFS differ between prokaryote and eukaryote. A. Two-team ribosome, polyribosome, simulate and translate messenger RNA. 70S initiation complex in a complex with elongation factor, and the peptide bond formed between the two adjacent amino acids. Translocation occurs as the ribosome moves one codon to the right, and when translocation is complete and the peptidyl transfer RNA is in the B site uncharged, and charged transfer RNA is released from the E sites. The elongation cycle repeats until stop codon is encountered, and once 70S initiation complex is formed, the FMT is bound to AUG codon in the B site of the ribosome. The cycle starts here in initiation complex. This elongation, the bond formed, translocation occur, and translocation complete, and elongation cycle repeat. And in the animation it appears clearly. This process we explain it in the animation, but this is a detailed process, step by step. 5 ribosome reading same RNA sequence, initiation codon, ground polypeptide chain, and complete polypeptide ribosome. Termination of translation signaled by stop codon. UAA, UAG, UGA stop codon have no corresponding transfer RNA. Release factor bind to stop codon and assists ribosomal in termination translation. RF1 recognize UAA and UAG. RF2 recognize UAA and UGA. RF3 stimulate termination. Termination events are triggered by release factor, peptidyl transferase enzymes that forms peptide bond. transfer RNA is released, ribosomal subunit and RF separate from single RNA. FMT or MET usually is cleaved from polypeptides. Stop codon is encountered and released. Factor bind to the stop codon. Polypeptide chains released. Component separate. And the animation, in the animations it appear clearly, but we try to explain the process in full detail. And further reading in the references, you can get more detail and more steps